share the slides. All right, folks, we're going to get started in under a minute here. I just want to remind everyone that during this session, if you have any questions for our speaker, you are welcome to use the raise hand reaction or to put that question in the chat. If you're feeling shy, you just want to put it in the chat, I will read it out loud to the speaker when it is time for their question and answer period. And both putting it in the chat and raising your hand are both completely unobtrusive. So you don't have to hold your questions till the end, but we will receive them in the answer that they were issued. So without further ado, we're going to move straight into our session. We're going to be hearing about evidence of convergent evolution in the nuclear and mitochondrial genes across deep squamate lineages. All right, take it away, Oscar.
Yes, him. Okay. Good morning, everyone. I'm Oscar Vannhofer, PhD student from the University of Bologna and member of the Molecular Zoology Research Group. Today we'll talk about mitonuclear evolution in the squamata clade. I cannot, okay, thank you. Okay. Squamatan shows a case of deep mitonuclear discordance, in particular the monophyletic origin of Iguania, constitute of Clerodonta and Acrodonta, is reacted using mitochondrial markers. That instead suggest a close sister relationship between Acrodonta, in particular between the Agamids family, and snakes, the current group of snakes. This sister relationship was at least partially explained, suggesting convergent evolution between these two lineages on the OXPHOS system, so on the oxidative phosphorylation pathway. This metabolic pathway is involved in the production of energy, so it's particularly important for fitness and take place in mitochondria. The OXPHOS system is composed of 5 complexes, and with the exception of complex 2, the other 4 consist of both mitochondrial and nuclear subunits. Mitochondrial OXPHOS usually constitute the core of each complex, while nuclear OXPHOS act as ancillary proteins and form a sort of shell all around the complexes. A subset of nuclear OXPHOS called contact nuclear OXPHOS tightly interact with mitochondrial counterparts. Literature posits coevolution between mitochondrial and nuclear OXPHOS to ensure a proper functioning OXPHOS system, so electron transport chain and the ATP synthesis. Here we test 3 questions. The first question concerns the phylogeny. So it's possible to retrieve the mitochondrial topology, so the discordance, also using nuclear OXPHOS genes, and how do mitochondrial and nuclear OXPHOS co-evolve in squamata. The third question concerns the hypothesis of convergent evolution between agamids and snakes on the OXPHOS genes. It's possible to detect some signals of this convergence in nuclear OXPHOS that was already demonstrated in mitochondrial subunits. Our dataset consisted of 56 species, and we created 3 gene datasets, a set of BUSCO genes that we used as control, and mitochondrial and nuclear OXPHOS genes. Firstly, Firstly, we performed a phylogenetic analysis, and we compared the topology obtained using BUSCO genes with the ones obtained using mitochondrial subunits, mitochondrial genes. We confirmed the literature, so we retrieved the discordance, and here you can see the sister relationship between Pinocephalus versicolor and Pogonavis viticeps, our two gametes, with the crown group of snakes. The nuclear oxphos maximum likelihood tree support a hybrid topology. We did not retrieve the monophyletic clade of iguanians, but neither the mitochondrial topology that was supported by only the 19% of quarters, according to the likelihood mapping analysis. But when we restricted the analysis to the contact nuclear oxphos, we retrieved exactly the mitochondrial topology. And here, the 88% of quarters support the sister relationship between snakes and agametes, so the black lizard in this plot. That result was coherent with the evolutionary rate correlation analysis, commonly used approach to investigate about protein interactions and coevolution. It's based on the idea of correlation between branch lengths of different set of genes. Here we retrieved positive correlation between mitochondrial and nuclear oxphos with a Spearman's error of 0.65. To test the robustness of this result, we normalized our dataset using 1,000 of randomly extracted subset of BUSCO genes. And then we plot, as you can see in the box B, the curve of distribution of Spearman's rho of all the correlations between, for example, nuclear oxphos and mitochondrial oxphos, the blue curve that had a median of 0.65. The yellow curve represents instead all the correlation between contact nuclear oxphos and mitochondrial, and it is significantly higher. In the background, there are the two contrast curves resulting from correlation between mitochondrial oxphos and BUSCO genes and nuclear oxphos and BUSCO genes. And what about convergences? Here, for each mitochondrial and nuclear oxphos, we reconstructed all non-synonymous substitution along the spermata topology, creating tables like this. Then we listed for each pair of branches the number of convergent substitution and the number of total substitution. These two columns were used here in the box 3. That's an example of what we've done on one gene. What was done using one gene. Each point in the graph represents a pair of branches, and its position resulting from the convergent substitution and total substitution between the two branches. We calculated the residual for each point, so the distance, the difference between the observed convergent out of total substitution and the expected ratio using a linear model. Using a density distribution of residuals, we highlighted the pair of interest, so the agamete snakes pair with the color red. Then the other gray point represent points within the confidence interval. For space reasons, we removed for each plot to plot the density distribution. In the next slide, you will see some yellow points. These points represent some pair of branches that we used as control. Nuclear oxphos, about mitochondrial oxphos, sorry. 11 out of 13 genes suggest strong evidences of convergent evolution between agamids and snakes. And since 11 red points are in the positive extreme of each of the distribution. About nuclear oxphos, we observed a similar pattern. 26 out of 75 nuclear oxphos reported the red point in the positive extreme. So nearly the 30% of nuclear oxphos show the same pattern of convergence. So my take-home messages are three questions. We detected the same phylogenetic pattern, the same phylogenetic signals between mitochondrial oxphos and at least a specific subset of nuclear oxphos, the contact nuclear oxphos. This effect is likely due to coevolution between nuclear oxphos and mitochondrial subunits, in particular between the contact ones. And the third take-home message concerns the convergent evolution. We detected strong signals of convergent evolution in mitochondrial genes, but also in nuclear oxphos. So oxphos subunits, so genes that interact with. Thank you.
All right, excellent talk. So we have time for questions. And just as a reminder, you are welcome to either raise your hand and we can unmute you, or you are welcome to put questions into the chat, at which point I'll read them out loud so that they're on the record. While we wait for people to put things in the chat, I have a question. So why do you think these two lineages show such strong convergence with one another? What do you think that is due to?
Sincerely, we don't know the real question, the real biological answer. However, it was already suggested that snakes have a particular physiological adaptation to their metabolic path, on their metabolic necessity of oxygen, because they may not food for, they do not eat for also long time between each meal, and it was suggested that agamids show a similar adaptive pressure, were under similar adaptive pressure along their evolutionary history. That's why they today that's it. I'm sorry, I'm a bit excited. That's my first talk. Okay.
It's just interesting because you think of agamids as being a fairly large, like that's a pretty diverse group in terms of their lifestyles and all of that sort of thing. Yeah,
because they may be under the same evolutionary pressure on specific metabolic pattern associated to high fluctuation, maybe in oxygen requirements or something like that.
Mm-hmm. That's very interesting. So what do you plan to do with this next?
Maybe study deeply the interaction between 3D models between mitochondrial and nuclear oxphos to try to detect some issues, some signals of real protein convergent in real 3D models.
Excellent. So you do have one question in the Q&A, which is this published? And could you share a reference in the chat if it is? So that's something for you to take care of after you talk.
Okay, yeah, I'm ready. No, it's not already published, but we are writing it. in the next month, I think it will be available.
Excellent. Well, thank you so much for your talk. I think we're about ready to get our next speaker loaded up.
Yeah. I am sharing my screen. So.
Yes. Thank you.
Thank you.
All right. Belen, you can go ahead and get your screen share going. Yes, our next speaker will be presenting in Spanish. If you would like, on your captions menu, so under more and then captions, you do have the ability to set the speaking language to Spanish and the caption language to English if you would like those, or if you would like the talk translated for you. All right, excellent. Those slides look beautiful.
I'm going to turn off the camera because the internet is a little slow.
Right, you're free to start whenever you're ready.
Okay. Can you see okay?
Yes, they look great.
Okay. Bueno, hola, gracias a todos por estar acá. Mi nombre es Belén Batistela y les voy a hablar acerca de la genómica de conservación de las lagartijas Liolaemus del fin del mundo. Es un estudio realizado, bueno, como ya dije, en el famoso fin del mundo, en Argentina, que analiza las especies de lagartijas, de las que se tiene muy poca información hasta el momento, y a las que nunca se les ha aplicado estudios de genómica de conservación. Bueno, antes de empezar, quiero aclarar que soy una alumna de grado y voy a presentar mi proyecto de tesis, así que no tengo resultados todavía, y obviamente que estoy abierta al final de la charla a escuchar comentarios o recomendaciones que sumen a este proyecto. I'll say this in English too. I am an undergraduate student and I'm going to present my thesis project. So I still don't have results, but of course that I open to listening to comments or recommendations that, at the end of the talk that add to my project. Bueno, yo estoy estudiando en la Universidad Nacional de Cuyo, que se ubica en Mendoza, Argentina, tierra del sol y del buen vino, y que tiene el premio por el mejor Malbec del mundo. Bueno, para empezar un poco en tema, vamos a hablar de la genómica de conservación, que estudia las fuerzas del cambio evolutivo sobre las poblaciones a través de los datos genómicos. Es un campo nuevo que hoy en día nos permite responder preguntas relacionadas a amenazas sobre la biodiversidad y predecir la probabilidad que tienen las poblaciones de sobrevivir a cambios ambientales. Y esto se logra en gran parte gracias a las nuevas técnicas de secuenciamiento que hay disponibles. Bueno, aplicado en el campo de la conservación, esto nos da un poder estadístico sin precedentes, y esto es gracias a las bases gigantescas de datos que se utilizan. Y esto no solo permite, como ya dije antes, analizar posibles amenazas sobre la biodiversidad y predecir la probabilidad de que las poblaciones sobrevivan a ellas, sino que nos permite analizar la interacción que hay entre las variantes fenotípicas y el ambiente, gracias a la obtención de información precisa, tanto de las variantes genéticas adaptativas como de las estructuras poblacionales. Esto a su vez nos permite estimar modelos demográficos, detectar si un cuello de botella es reciente o es antiguo, e identificar qué adaptaciones serían claves para la supervivencia de los individuos ante cambios ambientales como lo son la destrucción del hábitat, el cambio climático y la contaminación. Bueno, dentro de la familia Liolamidae encontramos al género Liolaemus, que actualmente contiene 290 especies descritas, siendo reconocido como el segundo género de amniotas más diverso del mundo. Pero en los últimos 15 años se han descrito numerosas especies nuevas del género Liolaemus, y según las predicciones moleculares, podría superar en número de especies al género que actualmente es reconocido como el más diverso, que es el género Anolis. Bueno, dentro de este género tenemos al subgénero Eulaemus, que se encuentra presente en Chile, Bolivia, Perú, Uruguay, Brasil y Argentina, habitando ambientes muy diversos y muy extremos. Dentro de este subgénero encontramos a la sección Lineomaculatus, que es reconocida y famosa por incluir a los lagartos del fin del mundo, que es la especie Liolaemus magellanicus, y es la única especie dentro de toda la herpetofauna que es capaz de llegar al sur de la Patagonia Argentina, específicamente a la provincia de Tierra del Fuego. Bueno, ahora sí llegamos al grupo Kingi, que es el grupo que yo voy a estudiar, que contiene 14 especies de lagarto que están distribuidos exclusivamente en la Patagonia Argentina. Para que conozcan un poco y se imaginen, no se tengan que imaginar cómo es, la estepa patagónica es una ecorregión que se ubica principalmente en el centro y en el sur de Argentina y se caracteriza por tener un relieve de mesetas y valles y una vegetación baja, dispersa, dejando mucho suelo desnudo, como se ve en las imágenes. La vegetación es principalmente de arbustos y todo esto está adaptado al clima frío, seco y ventoso que hay. Bueno, la historia geoclimática de la Patagonia es compleja, ya que entre el Mioceno y el Pleistoceno sufrió reiterados ciclos de glaciación, donde durante la época de glaciación el permafrost predominaba en el suelo, pero algunas pocas zonas, por ejemplo las áreas cercanas a los lagos, ejercieron de refugios filogeográficos para los individuos de este género, ya que eran zonas que presentaban un clima más ameno y que permitía la supervivencia. Después, en los periodos interglaciares, cuando el hielo se derretía y el frío disminuía, se habilitaban mayor cantidad de áreas con condiciones ambientales aptas para que pudieran sobrevivir. Estos cambios podrían haber afectado la dinámica poblacional, ya que se generaron épocas de aislamiento, seguidas por épocas de contacto secundario. Bueno, como ya se sabe, los reptiles son un taxón altamente vulnerable a los cambios ambientales, ya que al ser ectotermos dependen de la temperatura ambiental para mantener su temperatura corporal. Y aunque esto es cambio climático, es algo que se aplica a todos los lagartos, las características ambientales particulares de la región que habitan, o sea, de esta estepa patagónica, los hace aún más susceptibles a estos cambios ambientales abruptos. Según la USGS, todas las especies pertenecientes al grupo Kingi se encuentran dentro de la categoría Least Concern, o sea, de menor preocupación, pero hasta el momento no se han hecho estudios genómicos para verificarlo. Por eso este estudio constituye un aporte valioso de información en ese sentido. Algunos de los objetivos que se plantearon son el realizar un análisis bioinformático intensivo utilizando los datos genómicos de repositorios abiertos de especies de lagartijas del grupo Kingi. Y esto se va a aplicar por primera vez en estudios de genómica de conservación, comparando la variabilidad genética entre las diferentes poblaciones. Y un segundo objetivo es incluir un análisis de modelado de nicho para predecir la disponibilidad de nicho a futuro, dado escenarios de calentamiento global. ¿Cómo se va a hacer? Rápidamente. Para el trabajo se va a utilizar la base de datos genómica más grande disponible hasta el día de la fecha para el grupo Kingi, el cual va a incluir el genoma de 159 individuos colectados en la ecorregión patagónica argentina durante más de 20 años de trabajo de campo. Estos datos fueron obtenidos bajo la técnica RAT-seq, tanto pair-end como single-end. Cabe aclarar que cada lugar de colecta de individuos fue georreferenciado y estos datos se van a usar posteriormente. En segundo lugar, se va a obtener un ensamblaje de novo preciso para después realizar una curación con el objetivo de asegurar la buena calidad de los datos genómicos. Y en esta curación se va a hacer un filtrado para eliminar los indels y se van a exportar estadísticos de calidad. Después se va a analizar la ancestría para definir las poblaciones que se van a utilizar para el cálculo de estadísticos. ¿Y cuáles son los estadísticos que se van a calcular? La estimación de Pi para analizar la variabilidad genética, Tajima para analizar la expansión y contracción poblacional y la heterocigosis para cada población. Además, teniendo en cuenta que los periodos glaciarios e interglaciarios que ocurrieron en la zona podrían haber generado ciclos de aislamiento y posterior contacto secundario, como ya dije, se va a ajustar un modelo de aislamiento por distancia para poder analizar en detalle cómo la distancia geográfica estaría afectando el flujo entre las poblaciones. Por último, pero no menos importante, se va a realizar un modelado de nicho con el objetivo de predecir el rango geográfico que podría ocupar el grupo Kingi en un futuro. Esto nos permite identificar qué especies corren mayor riesgo de extinción o qué zonas se deberían priorizar y proteger. Para construir esta distribución se va a usar primero los puntos de colecta georreferenciados que nombré anteriormente, y luego las capas climáticas descargadas de WorldClim bajo escenarios de futuro calentamiento global. Bueno, para el trabajo informático se va a utilizar el clúster TOCO, que está ubicado en la Facultad de Ciencias Exactas y Naturales en la Universidad Nacional de Cuyo, en Mendoza. Bueno, algunos de los resultados que esperamos son, en primer lugar, que las poblaciones que se encuentren más distanciadas geográficamente tengan una mayor divergencia genómica entre ellas que las poblaciones más cercanas. En segundo lugar, que las poblaciones que habiten áreas que actuaran como refugios durante las épocas de glaciación tengan tasas de heterozigosidad más alta y una mayor variabilidad genética que aquellas poblaciones jóvenes que han recolonizado nichos más recientemente. Y por último, el modelado de nicho mostraría que las áreas de distribución de estas poblaciones se va a reducir y se va a desplazar hacia zonas del sur o de mayor elevación para compensar el aumento de la temperatura que está generando hoy en día el calentamiento global. Bueno, ya que hasta el momento para ninguna de las especies del género Liolaemus se han realizado estudios de genómica de conservación, los resultados podrían ofrecer información valiosa que respalde posibles planes de manejo o posibles áreas prioritarias de conservación. Y además, con la información obtenida, se podría recomendar modificaciones a la USCN acerca del verdadero estado de conservación de las especies pertenecientes al grupo Kingi. Bueno, para terminar, quiero agradecer al grupo de herpetología patagónica, especialmente a Melissa, Kevin y Mariana, que me insistieron para que participe acá y me escucharon 1000 veces mi exposición para que practique. Así que muchas gracias, y gracias, bueno, a Aliaisa y a Lipec, que son los institutos donde estoy realizando mi tesis. Y gracias.
Excellent talk. Thank you. At this point, we do have time for questions. And so if you would like to ask a question to our speaker, you can either put that in the chat and I will read that out, or I can allow you to unmute. All right, it looks like we already have a question, so I'm going to go ahead and ask that question. Ask her to unmute and ask it.
Should I do the question in English or in Spanish?
It's the same, if you want.
Okay.
Igual, I can see the captions. Por las dudas, hablemos en español, pero debería entender.
Ok. ¿Es posible tener en cuenta también como la capacidad de dispersión de estas lagartijas para que puedan migrar en un escenario de cambio climático? O sea, que de pronto el cambio climático es muy rápido y no los deja moverse lo suficiente como para no extinguirse.
O sea, las 14 especies están distribuidas en prácticamente toda la Patagonia. En este caso yo estoy teniendo en cuenta la dispersión con respecto solamente a las zonas de refugio, pero sí, yo en mi tesis no lo estaría teniendo en cuenta, pero se podría tener en cuenta.
Okay, excellent. Let's see, do you have a follow-up question, or are we open to take another one? Okay. Well, does anybody have any additional questions for our speaker? So I'm a little curious, towards the end of your talk, you mentioned making conservation recommendations. Is that something that you see yourself as interested in doing in the future?
Sorry, can you repeat my internet? It's a little slow.
Oh, no problem. So you mentioned making recommendations to the IUCN and conservation. Is that a role that you see yourself as fulfilling? Is that something in your future?
Yes.
Yeah. Excellent. All right, well, let's move on and get our next speaker up. I'm going to stop your share so that Danielle can go ahead and start hers. Perfect, we can see those just fine.
Okay, great. And I apologize in advance. There's a massive thunderstorm outside of my window all of a sudden, so hopefully I will stay connected. But thank you for joining to hear about one of my recent projects looking at how bioluminescence and environmental light influences visual evolution using a deep sea shrimp system. And for those of you that aren't so familiar maybe with marine systems or deep sea systems, light really does function as the universal language in these habitats where light can be very limiting. So light is attenuated with depth. And at about 200 meters, you're really only getting blue wavelengths of light that are coming through the ocean layers. And by about 1,000 meters, that light is completely absent. And really only point sources of bioluminescence are present at that point. So light has been shown to drive sensory system evolution in these marine habitats and really plays a fundamental role in behavioral responses of the animals, their overall fitness and survival, as well as their life history. So visual systems previously for a lot of these deep sea organisms have been thought to be limited to blue light vision. And again, this is because only blue wavelengths are really passing through past 200 meters, which is what we defined as the deep sea. And this is kind of opposed to, other terrestrial systems pictured in this figure where animals are thought to have, a number of different photopigments maximally sensitive to different wavelengths of light. So for a majority of deep sea fish, cephalopods, and crustaceans, it's really been historically thought that they really just have one visual pigment, maximally sensitive to this blue light. And again, I already discussed, the types of light that can be found in this environment and how limiting it can be, but both downwelling light and bioluminescent light typically, even though it's not emitted at the same exact wavelength range, it's typically emitted as blue light around 460 to 490 nanometers. But since, this first, was hypothesized about deep sea animal vision, there have been various exceptions found across deep sea taxon, where we're seeing that they actually have a diverse visual response. So there have been various exceptions found in fish, as well as in vertically migrating deep sea shrimp. So these opal-fored shrimp that I study, they're now believed to have at least 2 photopigments maximally sensitive to those blue wavelengths of light, but also, UV shifted light, so shorter wavelengths of light. And since, these physiological studies have come out to show that they actually likely have at least two photopigments, various studies by myself and some of my colleagues have been able to show that additional species within this shrimp family, these opal-fored shrimp, appear to have even more diverse visual options than previously thought. So not just blue or UV shifted short wavelength options, but they also appear to have long wavelength green sensitive options as well. And those options are kind of the major protein that are making up those photopigments that I mentioned. And in a recent paper by one of our colleagues in 2024, he was actually able to show that various of these options that we recovered are actually functional. So another interesting thing about these opal-fored shrimp is that they participate in this diel vertical migration in the ocean. And this phenomenon can be found across aquatic environments, and it's basically where animals from deeper waters swim to shallower waters at night to feed, and then return to deeper, darker waters during the day to hide from visual predators. And this schematic here is showing you some acoustic data, kind of showing you over time how those animal kind of masses change density with depth over time. And collectively, this represents the largest migration on Earth. So a lot of animals are doing this. And during this time, these animals are passing across, large kind of ranges in depth, which come with their own environmental conditions. So they're subject to a variety of environmental conditions while they are migrating. And this is particularly true for these deep sea vertical migrators that I study that migrate hundreds to thousands of meters daily to actually feed and mate in shallower waters. So they're really experiencing drastic changes in environmental variables, including light, temperature, and pressure, which can have really serious impacts on sensory systems. But the overall influence on visual evolution is still poorly understood. And it's not very easy to study this phenomenon and these animals. So we're trying to do this in various ways. And our more recent project is focusing on looking at visual evolution across the superfamily Oplophoroidea. So these are Caridian shrimp, and they are comprised of two main families, Acanthophoridae, which has 7 genera, and Oplophoridae, which has 3 genera. And there's about 70 accepted species, and they all inhabit very distinct kind of and variable depth zones in the open ocean. So from the mesopelagic, bathypelagic, or benthopelagic zones in the ocean up to about 5,300 meters. So really diverse and versatile group here. And many species within this superfamily participate in this vertical migration that I mentioned, but the extent of that migration can vary greatly across the superfamily. And just an example of this is one species, Cystilaspis debilis, is considered a strong migrator. So it can migrate, from 4,600 meters to about 150 meters, whereas Ephrina umbango is a weak migrator and stays relatively deep. So, where light is very limiting. Another interesting thing about this superfamily is that they all are presumed to have these bioluminescent oral secretions. So they're all bioluminescent, but certain strong migrating genera within one of the families, Oplophoridae, has evolved a secondary mode of bioluminescence in terms of these light organs or photophores, which just as a side note, we were able to show function actually as extraocular photoreceptors, but they're believed to primarily aid in camouflage and counter illumination, kind of distorting the body profile in the water column as they migrate. And it's really these photophore-bearing oplophorid shrimp that had previously been shown to have, at least 2 photopigments. So they appear to have an expanded, visual diversity. And we believe it's possibly to help them discriminate between light sources, so maybe downwelling light and bioluminescence in the ocean. But we know that light is functioning as a driving force for visual evolution in the deep sea. And the kind of molecular basis for this light detection, as I mentioned previously, are these opsin proteins. And kind of the structure of these proteins dictates, what wavelength of light that protein is sensitive to. And this really comes down to the amino acid composition. So we can study this at the molecular level. And by having a higher number of opsins and a higher number of types of opsins, so diverse opsins, you can have an expanded photoresponse, potentially see light. This typically happens through gene duplication and divergence events. So for this study, we wanted to characterize opsin diversity across this superfamily to, again, really understand the influence of these different light sources on visual evolution, selecting species with both single and dual modes of bioluminescence and a diverse range of migratory behaviors, and then try to estimate the ancestral opsin repertoire of this superfamily to better understand trends in opsin gains and losses in more of a phylogenomic context. And then look for evidence of positive selection among opsins and other eye genes in those photophore-bearing species to see, if this evolution of this dual mode of bioluminescence, which might be functioning potentially in communication with conspecifics, may have, exerted strong selective forces on visual evolution in this group. So I'm going to briefly just go through these methods for the sake of time, but we were able to collect various species across the superfamily, preserve them in RNA later. I was able to dissect out the eye tissues, extract RNA and sequence the RNA from 11 species within the superfamily, including one outgroup. and various replicates. I was then able to sequence the transcriptomes of these eyes, sequencing data, and identify orthologous genes within these transcriptomes so that I could build a tree for the superfamily Oplofuroidea, which comprised 622 orthologs. And to kind of get at this first objective to characterize visual opsin diversity across this group, I was able to use phylogenetically informed annotation tool PIA to actually annotate and curate those visual opsins and then map them back onto this phylogeny I was able to create for the superfamily. And this phylogeny, this tree here, is the result of that, again, built with 622 orthologs. The opsin diversity that was characterized for each different species in this tree is represented here, with SWS representing different distinct short wavelength sensitive opsins, MWS mid wavelength sensitive, and LWS long wavelength sensitive. So kind of absence of color here indicates that that opsin was not found across that species. So kind of the main takeaway is opsin diversity was highly variable across this superfamily, and that very diverse within our one family represented by this top plate here, Acanthophiridae, which appears to be more of a deeper dwelling family, as opposed to the family that has evolved that dual mode of bioluminescence in the shape of those photophores that I mentioned, and they tend to be shallower. To estimate the ancestral opsin repertoire, I used ancestral state reconstruction. And those reconstructions are represented here, again, using presence absence of the opsin types to calculate probabilities that those opsins were present in each common ancestor across this tree. And this is represented at select nodes here. And really the main takeaway is that the ancestor of this superfamily had an expanded opsin repertoire, including short wavelength, mid wavelength, and long wavelength sensitive opsins, which is similar to what's been predicted for the ancestor of vertebrates as well. And then we were also able to do ancestral state reconstruction for the depth ranges of these different animals across the superfamily, really to show that the common ancestor, again, of Oplofuroidea was likely deep dwelling, and this is represented by the pie charts here, whereas the common ancestor of the family that evolved those photophores appears to have been a shallower migrator. So potentially migrated to shallower photic environments where those photophores would have been, providing some type of selective advantage. For this kind of third objective, for the sake of time, I'll run through this quickly, but I was able to test for positive selection across these opsins as well as other eye genes using CODA-MEL. Again, really focusing on those photophore-bearing species to see if their opsins were under selection. And I was able to identify positive selection for one of those mid-wavelength sensitive opsins, particularly at sites that are interacting with the retinal binding pocket, which kind of changes the chemical environment around retinal, which can lead to altered spectral sensitivities. And this mid-wavelength sensitive opsin is believed to be sensitive to blue light. So, it's possible that diversification of this opsin has facilitated spectral tuning among oploforids and actually is enabling them to differentiate between light sources in the ocean, potentially between photophore emissions of conspecifics. So just to quickly summarize, we were able to characterize opsin diversity across Oplofuroidea to show that the common ancestor of Oplofuroidea had a diverse opsin repertoire, which challenges that paradigm that vision in most deep sea species is limited to blue light. And then to show that there's selection acting on a mid-wavelength sensitive opsin in our photophore-bearing species, possibly enabling light source discrimination in the oceans. And with that, I just want to thank my lab, the Chrostomics Lab at Florida International University, and our funding sources.
All right, excellent talk. Do we have any questions for our speaker? As a reminder, in case it's your first time. Okay, here we go. We've got one. Let me invite our question asker to unmute.
Hi, great talk.
Thank you.
Yeah, so I was wondering, what test did you use to identify signature of positive selection?
Yeah, so I used various pairwise model comparisons. So I did both branch site and branch site model comparisons, all of which came back significant for that mid-wavelength sensitive opsin. I was also able to run other programs to test for evidence of selection. So I also ran Meme and Hyphy within different software packages as well, which also came back with positive results for this one particular type of opsin. We're also able to identify other genes within those I-transcriptomes that are under selection, but it's a little too much to fit into this talk.
Amazing, amazing. So there were any in those tests, were there any like amino acid substitutions which indicated, or it was the McDonald-Creatman test? Like I was just curious to know that, like, what signature actually convinced you that there is positive selection. And I might not be familiar with all the softwares that you are using, but yeah.
Yeah, so
Actually, can
sorry.
I have you guys take this into the checks? It is 1:15,
Sure.
which means we have to move on to our next block.
Absolutely. Thank you.
Thank you so much. All right, Hannon, are you here? You can go ahead and get your slides up. There we are. Hello. Here we go.
Okay, can you see that?
Yes, I can.
Awesome. Thank you for having me.
Whenever you're ready, you can go ahead and get started.
I'm going to present some work today done in collaboration with Joanna Maisel and members of her lab. I'm a grad student at Stanford University. And our work was motivated by this prediction of Tomoko Ohta's, sort of the central tenet of the nearly neutral theory, which is that there will be variation among species in the ability of selection to purge weakly deleterious or fix weakly beneficial mutations. And specifically, Ohta supposed that this would vary with effective population size. So when effective population size is small, selection will be less effective. That is, weak mutations will be effectively neutral. When effective population size is large, selection is more effective, and even mutations with very small effects will be able to be fixed or removed on average. And this hypothesis has been borne out by a number of empirical studies. Specifically, genome size varies with the effectiveness of selection. When selection is more effective, genomes are smaller and more streamlined, and so does codon usage bias. So there's a greater degree of bias in which codons are used for an amino acid in species where selection is more effective. And so we asked simply whether the amino acid composition of a species proteome also varies with the effectiveness of selection. And amino acid composition is a pretty coarse measurement, right? You're looking across the whole proteome, but it actually corresponds quite closely to physical properties that might interest us in the proteome, like secondary structure, disorder propensity, pKa's, things like that. And so we used three methods to tackle this question. The first of which is a maximum likelihood method. So across these two clades, Primatomorpha is primates and sister Colugo, and Glirase is rodents and rabbits. We fit a non-stationary model of amino acid evolution by simply holding the exchangeability matrix constant, but allowing amino acid frequencies to vary. So we're estimating equilibrium max likelihood frequency for primates, which we know tend to be low and decreasing any, and Glirase, which we know tend to be high, and over this tree increasing any or effectiveness of selection. And this allows us to calculate the flux in amino acid for each amino acid from the root to primates, from the root to glirase, and thus the differential as well between primatomorphs and glirase. The second method zooms in on 4 species within that tree for which we have very good polymorphism data, and uses parsimony to calculate amino acid flux at each site. So for sites that have a pattern like this, where a single species has a different amino acid to all of the others, we infer by parsimony that the ancestral state was what the other species had, i.e. here that on the rat branch, there was a transition, a substitution from valine to alanine. And we can count up these sorts of patterns and estimate for each amino acid whether it tends to be in the derived state or the ancestral state more often, i.e. its flux on each of these 4 branches, terminal branches. And this method is not new. It's been used for single proteins and proteomes before, and we should talk in the discussion about Jordan et al.'s results and why they may be wrong. And then finally, we zoomed out from that glirase and primatomorph tree to look across vertebrates in case to see if our results were due to the biology of those organisms in particular, or the differential we're trying to measure between high NE and low NE. And we measured codon bias, which we know is correlated with the effectiveness of selection, or product of the effectiveness of selection, versus amino acid frequency. Codon bias is measured as the codon adaptation index of species, which is corrected for amino acid composition and GC content. And so we have codon bias, a measure of the effectiveness of selection on the X, and the frequency of an amino acid on the Y. And so we can see that for lysine, species with greater codon bias, i.e. more effective selection, have less lysine. For glutamine, there is no such relationship. And for arginine, for example, species where selection is more effective have more arginine. And so for each of the 20 amino acids, we measure this relationship. And that gives a sense of, at the whole vertebrate taxon scale, what's going on. So I'm going to go back to the maximum likelihood results just in the primatomorphs and glirase tree, and show you that indeed some amino acids on the Y here are gained, are at higher frequency in rodents than in primates under this maximum likelihood estimation. So like proline and alanine up here we see. And this relates to a lot of really interesting biological properties. So rodents prefer these amino acids that are cheaper in terms of the ATP needed to synthesize them, and also those that are not dietarily essential. They also prefer smaller amino acids, and those amino acids that have a tendency to be in disordered parts of proteins. And rodents versus primates prefer amino acids that also tend to have a benign effect when you express them in E. coli. This was somebody else's experiment, but we saw this observation in random peptides, so agnostic to protein function, just looking at the effect on growth. And finally, we see that rodents over primates tend to prefer amino acids that have greater percent GC in the codons that encode for them. And this makes a lot of sense, right? Because the mutation biases that we best understand, the mutational processes that we best understand have a bias towards AT. And so under more effective selection, apparently we see that rodents are able to overcome that AT bias and maintain those high GC amino acids. Now I'm going to compare methods. So you're seeing that maximum likelihood glirase versus primatomorphs on the bottom. And then on the Y, we have the pan-vertebrate measure of whether an amino acid is at higher frequency when codon bias is higher. And we see broad agreement between these two. Now I'm going to bring in the parsimony method just in those 4 species. And so those amino acids that are preferred across rodents versus primates by ml are also preferred in mouse and rat versus human and chimp. And then those amino acids that are preferred in human and mouse and rat versus human and chimp are also preferred across vertebrates. So the three metrics broadly agree on which amino acids are either gained or at higher frequency under more effective selection, or when selection becomes more effective, depending on the metric. And you can see that not just across the 20 amino acids, but also within pairs of highly exchangeable amino acids in the BLOSSOM and PAM matrices, there is a pretty strong degree of preference. So looking at R and K and I and V on here, here are the structures of those amino acids. So you can see arginine and lysine are both positively charged. They're similar in a lot of ways, but arginine is far preferred. And similarly, valine is preferred over isoleucine, even though they're chemically quite similar. And interestingly, we see a preference or an increase in R and specific K to R substitutions in transitions to life at high temperatures in animals, fungi, and prokaryotic taxon. Finally, using that parsimony method where we're looking at site-wise, we can look at each substitution that we could infer occurred. We see that in rodents, looking at that lysine to arginine pair, not only does arginine increase and lysine decrease in frequency, but that observation is driven by specifically substitutions from lysine to arginine. And similarly, isoleucine to valine substitutions dominate the isoleucine and valine landscape. But for primates, where selection is less effective, we don't see that process occurring. We see a lot of other stuff going on, a more diffusive process, maybe where selection is less effective. And then finally, in rodents, where we see this strong preference and where we see the same preferences across vertebrates, across the glue race, why would you care between these amino acids? This is something that needs testing experimentally, but our hypothesis for the time being is that lysine and isoleucine, the two that are lost, have more degrees of rotational freedom. There are more carbon-carbon bonds that have full rotational freedom. And so you can imagine as you fold those proteins, you restrain those bonds, they can't rotate as freely, and you have a greater entropic penalty. You lose entropy when you fold, and that's a bit less stable. So it could be that what we're seeing when selection is more effective is selection for greater free energy of folding. That is, we're seeing sort of the knife edge that marginally stable proteins sit on in the balance between mutation, selection, and drift. And so I'd like to thank Joanna and all the members of her lab who helped me with this project, people we've talked to, and my graduate advisor, Paula Wielander, for letting me do this. And you all for listening, and I'll take any questions.
Excellent talk. Thank you very much. So at this point, we have time for questions. We do have enough time to take a few. So if you would like to be acknowledged, please raise a hand and I can ask you to unmute. Otherwise, you are welcome to put those questions into the chat. So I'm a little curious actually about this. I'm more on the inference side of, inferring phylogenies. And, a lot of our models for inferring phylogenies are based on having some, biochemical idea about how mutations happen. Do you see these results as potentially not just impacting, what we can assume about molecular evolution, but how we could infer phylogenies?
Absolutely. Yeah, I think I interpret these results as a pretty strong argument for non-stationary models for evolutionary inference whenever possible, because you just see the change in amino acid frequency, probably also change in exchangeability, but at least frequency over time. And so wherever we can model that realistically, I think it'll do a better job.
Yeah, that's interesting, and especially over the depth, because a lot of times people talk about the non-stationary processes with the really deep nodes, but you were showing that even in fairly, shallow ones, this could be a huge issue.
Yeah. And we were the only thing that was not stationary in our model was the frequencies, and that already helped a lot. So yeah, if there's not a lot of data sets big enough maybe to let the exchangeabilities vary, but if we could somehow model that, that would be very cool.
Yeah, interesting. Excellent. So we've got to get our next speaker up. So if you have any further questions, Hannon will be here in the chat. Let's see. Our next speaker has asked not to be recorded, so we are going